This recorded session is dedicated to our lecture on cell cycle. We're going to continue our discussion on cell division. And for this part, or for this recorded session, we will cover meiosis, which is a unique process of nuclear division observed in gametes, in sex cells. So, unlike mitosis, meiotic division is fundamentally different in terms of the preservation of the diploid state of the parent cell, or the inheritance of the diploid state of the parent cell by the progeny cells. So you will remember from our discussion last week that after mitosis, we expect the daughter cells to be genetically the same with the parent cells, and the number of chromosomes are still the same. So if the parent cell has a total of 100 chromosomes, then you expect the daughter cells to have the same number as well. You know? So unlike mitosis, in meiosis, there is a reduction of the ploidy from diploid to haploid. Okay, so um, in meiosis, or the meiotic cycle, the meiotic process is divided into two stages. So there is meiosis 1 and meiosis 2. So meiosis 1 is when homologous pairs of chromosomes look for one another or find one another. They synapse. So material, genetic material, or DNA material is recombined, and then later on they separate. No, just similar with the usual mitotic cycle. And meiosis two is where the product, or the daughter cells from meiosis one further divide, okay, to produce two other daughter cells. So in total, you expect that after meiosis, there are four daughter cells. Okay? And these four daughter cells are haploid. They are unlike their parent cell, the parent initial parent cell, which is diploid. Okay, so in this illustration, you can appreciate that from a total number of four chromosomes, the daughter cells are now possessing only two chromosomes each. Okay, so from a ploidy of from a diploid state, 2n of 4, the haploid state is achieved after meiosis 1 and proliferated, or maintained after meiosis 2. Okay? So, unlike mitosis, there are two distinct stages. So, there's meiosis 1 and meiosis 2. And there are important things to remember, or important processes to take note during the process of um, during the process of meiosis 1, because unique to meiosis, the interchange of the DNA genetic material between homologous chromosomes allow for genetic diversity. Okay, so you will expect, or you can expect, that the haploid daughter cells after meiosis 2 are different genetically. Okay, because there's recombination of sections of DNA, sections of genes during meiosis 1. Okay, so some of the important steps, or I'd say some of the stages I'd like to point out. So when a sex cell will start the meiotic cycle. So there are stages within prophase 1, where certain unique events happen. So, um, leptonema, so to enumerate, okay? The prophase 1 of meiosis 1 has several stages. So there's the leptonema stage, there's the zygonema stage, the pachinema stage, diplonema stage, and diakinesis. Okay, so let's take a look at some of the key events, right? So during the pachinema stage, right, or the pachytene stage, chromosome exchange or chromosomal material exchanges between the the maternal and the paternal homologous chromosomes, and that exchange is facilitated by crossing over. No, at the points of crossing over, bridges are seen um, uh, in between the homologous chromosomes, and that those bridges later on we'll discuss further will serve as as physical structures that facilitate the exchange of genetic material. So during pachinema, the homologous pairs of chromosomes have already established correct pairing with one another. Prior to pachinema, during the stage of zygonema and leptonema, there is what we call homologous search. So the homologous chromosomes will look for one another. Okay. Next, during the diplonema stage, okay, where you expect the threads of chromosomes of homologous pairs of chromosomes to be more visually appreciable. Okay, um, the the exchange of material, of genetic material, the crossing over event already took place, and those bridges, particularly the synaptonemal complexes, they will disappear. Okay, and the pairing of the homologous chromosomes will now be loosened or relaxed. Okay, so the pairing will now the pairs of homologous chromosomes will begin to be uncoupled or unpaired with one another. And then for diakinesis, right, the homologous pairs now at the a more loosened state, they will pull apart, okay, and there's already terminalization of the structures that were formed during pachinema, moving forward, and until crossing over took place. So those structures that were formed, they will now begin to disappear. Okay, so to facilitate the separation of the chromosomes that have now very different genetic material. Okay, now looking at meiosis 1, okay, you can expect the same events similar to mitosis, right? Then eventually, when the cell enters meiosis 2, it's just similar to a usual mitosis where the sister chromatids are pulled apart, okay, towards the opposite poles of the cells. So, or the cell to, separate or distribute the total number of chromosomes. Okay? So, how do homologous pairs of chromosomes look like? So, in this illustration, you can appreciate um, one homolog from one parent and another homolog from another parent, represented by two different colors. So there's blue and there's red. Okay, so during prophase 1 of meiosis 1, you expect homology search, homologous pairing, crossing over to take place between these between these pairs of homologous chromosomes. Okay. Um, I'd like to introduce the concept of karyotype or karyotyping, which is a visual representation of the chromosomes of a cell. No? In this example, in this illustration, you can see the karyotype of the human species, no? Of Homo sapiens. 22 pairs of autosomes, 22 pairs of of chromosomes that are responsible primarily on the um, non-sex contingent regulatory activities or elements. Okay, so from pair 1 to pair 22, those are what we call autosomes, while our sex chromosomes, X and Y, okay, that's a 23rd pair, X and Y or X and X, okay? The sex chromosomes, we call them our allosomes, okay? And they are responsible for for regulatory processes or elements that are contingent to the sex of the species, sex of the animal. Okay, another example of another illustration of a karyotype. So you can see different colors within a chromosome, okay, within a pair of chromosomes. So you can see similarity in terms of color. So those parts or sections or segments of chromosome pairs that are of the same color, we expect them to possess the same genes, the same set of regulatory elements or sequences that pertain to a specific function or pertain to a specific or that are involved for a specific function or a specific uh, regulatory mechanism or regulatory process, right? What else? Okay, so this is also another illustration of a karyotype. This time it's not really colored. Okay, mostly we can appreciate karyotypes to be in this format, black and white, but the banding of the color, the patterns between the patterns of chromosomal pairs are really the same. No. Okay, so this is, yeah, I mentioned this already: autosomes, somatic ones, and our sex chromosomes. Okay. So just as a review, all right. So during the homologous pairing, right, your one paternal chromosome and another maternal chromosome will find one another, form a homologous pair, right? Um, um, we expect them that we expect that after crossing over, they will separate, so they will be distributed, of course, to the progeny cells. So similar with the usual mitotic cycle or mitotic division, the same structures you can appreciate: the kinetochore, the centromere, the primary constriction in a chromosome, the presence of sister chromatids. Okay, so sister chromatids are chromatids from the same chromosome, from the same pair, while non-sister chromatids are chromatids that are essentially coming from, or physically we relate the chromatids from the paternal and the maternal sources. Okay. Now, an important structure that I want to discuss further is the synaptonemal complex. So, this is a key element in meiotic cell cycle. So, the synaptonemal complex, it's a protein complex. It's a multi-protein complex that is formed between homologous pairs of chromosomes during the prophase 1 stage of meiosis 1. Okay? And it's meiosis specific. You cannot expect it to be present or existing in mitotically dividing cells. Once that this synaptonemal complex is developed, it will mediate and maintain synapses along the full length of the homologous chromosomes, and um, the major function of the SC, of the synaptonemal complex, is for the maturation uh, of DNA double strand breaks that facilitate crossing over events. The exchange of DNA between the parental chromosomes, between the maternal and the parental chromosomes. So these crossing over event or crossing over events provide the physical connection that will hold the homologous chromosomes together until eventually they segregate at the end of meiosis 1. Okay? So here you can see the sequence of events from leptonema to from leptonema to diplonema, right? So from during leptotene, homology search is the key event. So the chromosomes, the homologous chromosomes will really try hard, will really ensure that they find one another. Okay. Then during zygotene, there is already rough pairing. So you can expect that homologous chromosomes, the complementary parts, have already paired almost completely. And during pachytene, the pachynyma stage, crossing overs to take place. Okay, synapses and crossing overs take place. Then afterwards, in diplonema, there's already relaxation of the paired sections, and homologous pair will now begin to dissociate. No, they will loosen up and be ready for distribution. Okay. So this is just a more illustrative example, right? So after meiosis, prophase 1 of meiosis 1, we expect there is recombination of genetic material. There is originally, so here you can see, originally your maternal and paternal chromosomes, they have very different genetic composition. So that's the difference is indicated by the color and by the uppercase or lowercase letters representing a particular trait, like for in this example. So here you can see that after crossing over, okay, there is genetic transfer, there's genetic material transfer in this section, okay. So you expect that later on, after meiosis 1, okay, the chromatids can be now called recombinant because there is unique combination there is crossing over and there's genetic transfer or DNA not transfer but exchange, DNA exchange, and the resulting chromatids have very different genetic constitution. So one will contain very similar to the paternal or the maternal, or two will be very similar to the paternal and the maternal chromosomes, and the other two will have more recombinant states. Okay. Right. Okay. So I've discussed this already. Now let's take a look at the process of gametogenesis, so the production of the sex cells, the female and the male sex cells, right? So the process of orgametogenesis as a process is unique between males and females. Okay, so males will undergo what we call spermatogenesis, while females will undergo oogenesis. And during those processes, the mature sex cells are the expected products, which are haploid in terms of their chromosome number. And during fertilization, they will combine, the egg and the sperm will combine and reconstitute the diploid state of the cell of the zygote. That zygote will of course become a new individual. Okay, so from haploid state, eventually the diploid state will be reconstituted. Okay, now a few things before we talk about spermatogenesis and oogenesis. Okay, so our sex cells, right? They come from what we call, they originate from what we call PGCs, primordial germ cells. Okay, so these PGCs, they are produced during our development, our early development, when after we have been after we come from the fertilization of an egg and the sperm that make up our that essentially are the um, unifying, or the donor cells that gave rise to the zygote. Okay, so these PGCs, is primordial germ cells, they have a diploid set of chromosomes. Okay, and they are precursors to the gametes. During embryonic development, these PGCs, they arise extragonadally, meaning they arise somewhere, not the gonads per se, not the ovaries for the females or the testes for the males, no? And they migrate to the genital ridges, so the presumptive genital ridges of the developing embryo. So please take note, okay, that as early as zygote development, okay, as early as the period where the zygote is continuing to achieve multicellularity, the origin cells of the gametes have already been produced. Okay, now these primordial germ cells proliferate via mitosis, and they reduce their number into half, but through meiotic nuclear division. Okay, and the final stages of maturation and differentiation of the gametes into spermatozoa or ova will have very unique characteristics, or unique processes. I think we can take a break to ensure that the recorded lecture is not too long.